Welcome to the Encore playback system. Please enter your conference ID number, followed by pound.
Welcome to the Encore replay of the Barclays Speaking the Science conference call series occurring on April 18, 2019 at 12 p.m. Eastern Time.
Is this the conference you want to hear? Press 1 for yes or 2 for no.
After the tone, please state and spell your first and last name, then press the pound key.
Vikram Khanna.
Recording completed.
After the tone, please state and spell your email address, then press the pound key.
Vikram at icariancapital.com.
Recording completed.
Good morning. My name is Catherine, and I will be your conference operator today. At this time, I would like to welcome everyone to the Barclays Speaking the Science conference call series. All lines have been placed on mute to prevent any background noise. After the speaker's remarks, there will be a question and answer session. If you would like to ask a question at that time, please press star and then the number 1 on your telephone keypad. If you would like to withdraw your question, press the pound key. Thank you. Ms. Gina Wang, you may begin your conference.
Thank you, Catherine, and thank you everyone for dialing in. We are very pleased to continue Barclays 2019 Speaking the Science conference call series. Our call series are intended to focus on key scientific concepts and the progress of preclinical and clinical programs. Please hold your questions until the end, but as always, feel free to email me at gina.wen@barclays.com if you prefer anonymously. Today on the call with me is Chuck Bond, President and Chief Executive Officer, and Jamie Crescenza, Executive VP and the Chief Scientific Officer from Mirati. Thank you, Chuck and Jamie, for joining us today.
Thanks for inviting us, Gina.
Happy to be here.
Sure. Thank you. And I know we have a lot to discuss about KRAS G12C today. So maybe I would just start with the big picture questions regarding KRAS G12C as an oncogenic driver. Given the preclinical data we have seen so far from multiple programs, including yours and others, do you still believe there will be a durable single agent activity for G12C inhibitors?
Yeah, I think I'll start and ask Chuck to add anything to this, but I think there are multiple lines of evidence that indicate that it's likely that a good inhibitor of KRAS would yield durable responses to inhibitor. I think in terms of background information, there's genomic and functional genomics data supporting KRAS being an oncogenic driver. Genomic data really indicates that KRAS is mutually exclusive with other known disease drivers like EGFR, ALK, ROS, and others, and is its own well-defined disease segment, and this is consistent with other oncogenic driver targets. Functional genomics data in large studies like Project Drive or Achilles have come up with KRAS in KRAS mutant cell lines. So just to give you a little bit of background on these studies, these are shRNA or CRISPR studies designed to knock down systematically every gene in the genome across hundreds of cancer cell lines. And really, one of the top scores to come out of this was KRAS mutant cell lines being scored high for the knockdown of KRAS, meaning that these cell lines are heavily dependent on KRAS. We extended these studies into our own shop, looking particularly at G12C. So at this point in time, we've looked at over 25 models. These include both cell line derived and patient-derived xenograft models. We're now extending this also into genetically derived models as well. And the outcome from these results indicates that around 75% of these models respond with greater than 30% tumor shrinkage. Indeed, a good subset of these models, perhaps around 60%, actually regress to the point of greater than 50%. So I think that's indicating that if these models are reflective of human disease, that it's possible that they would respond. Now, in terms of the durability, I think this remains a question that needs to be answered in the clinic. What we're doing currently in non-clinical studies is extending our mouse model studies out from the normal 30 days out to several months to determine if and when there's an onset of resistance. It's really too quick to early answer that question at this point in time. And then finally, I think if you look at the paradigm of oncogenic drivers and other molecules that KRAS signals through that are related to KRAS, BRAF is probably the closest corollary. Here, we believe that probably BRAF inhibitors like dabrafenib or dabrafenib plus trametinib are the best clinical surrogate for inhibitor activity of KRAS. And here, we recognize that the dabrafenib plus trametinib combination, which probably most closely phenocopies single-agent inhibition of KRAS, as this whole CRAF feedback loop will not be intact, has yielded response rates of over 60% and durabilities of around nine months. We think that that's probably the closest to KRAS. Now, just note here that there's a caveat. KRAS is a GTPase and not a kinase. We also believe that we need to cover KRAS entirely for the full dose interval, and having a compound that achieves that is going to be very important. This, too, is a question that's going to need to be answered clinically.
Yeah, and I think a couple of points just to emphasize that when Jamie says 25 different models, that's pretty extensive, as you probably realize, and you've looked across other programs. And so there aren't very many that have done that many different models. And we've seen consistent activity. So again, it indicates single agent activity and good, at least predictive of durable responses. And the other interesting thing, which probably isn't typical, is Jamie actually chose a number of cell lines that he thought would be resistant. So actually selecting the models against us, and that still showed good results. So I think that we're trying to answer this question as rigorously as possible, both about primary and secondary resistance, which I think we'll get to in further detail later as well.
Mm-hmm. Thank you. I think that's very helpful. Jimmy, you also mentioned, 25 different mouse models, and 75% had a tumor shrinkage over 30%, and then the other 25% did not respond as much. So do we, and then, you also mentioned, certain percentage had a much greater than 30% response, tumor shrinkage. So do we know, the differences or, heterogeneity of different tumors leading to different sensitivity to the G12C inhibitors?
Sure. First, I'll start by answering that with a clarifying point. So in these 25 models, we have seen significant anti-tumor activity in all 25. However, that ranges from seeing a complete or near complete response to some stable disease to some delayed progression. So it is around 20% of models where we only really see delayed progression or stable disease. In those models, we've studied, well, I will say across the 25 models, we've studied across the board all the genetic markers or any baseline expression correlates, either at the RNA or protein level. I would say here that there's not evidence of one single genetic marker that conveys resistance, but there are some non-significant trends in the models that we'll keep our eyes on. One of those non-significant trends is the observation that homozygous deletions of CDKN2A, which dysregulate the cell cycle, may trend more towards resistance. The other thing that we've observed is if you have upregulation of receptor tyrosine kinases in the HER family, that these are not significant, but they do tend to somewhat trend towards the more refractory models. So this is something we're not going to exclude patients from the clinical study based on these observations, just in that they're non-significant and they're also mouse models. However, also, I believe we're heartened in that if these do turn out to be models that trend a little bit more with less response, that we believe that they're both actionable through combination. The HER2 family, the HER family kinases being actionable through combinations with drugs like afatinib or cetuximab. The CDKN2A, we have some pretty compelling non-clinical data that the combination of our drug with palbociclib in that segment has yielded very strong anti-tumor activity, in some cases complete responses, in models that are pretty refractory to single-agent KRAS inhibitor or palbociclib. So going to continue to study this. We believe that these would be handles for combination strategies as well.
Mm-hmm. So are there any, specific tumor types that will be more, resistant or more sensitive?
It's hard to tell so far. We have evidence of lung, colon, and pancreas models responding. I would say the depth of response and fraction of responding models is likely strongest in lung and pancreatic cancers, whereas colon cancers are more of a mixed bag. But just to note, we really only have, out of these 25 models, around 4 colon cancer models. So we've expanded our modeling to better understand this and whether there are additional, there are additional factors in these colon cancer models that we can understand the likelihood of response. We do recognize that there's a history also with oncogenic drivers like BRAF inhibitors, where single-agent BRAF inhibitors yielded a lower response rate, likely due to upregulation of EGFR receptor tyrosine kinases. I think we're heartened again that there are combination strategies that will address this. For example, the Beacon studies that are reading out with RAF-MEK and cetuximab in patients with BRAF mutations have now yielded high response rates with very strong durability. And even though we may or may not see strong single-agent activity in the colon cancer setting, we believe that this precedented data provides a likely paradigm for KRAS to be able to develop rigorous combination strategies to interrogate that disease segment.
Mm-hmm. Okay. And then also, is there any scenario that KRAS G12C mutations develop, secondary or subsequent to a first driver mutation?
Sure. As mentioned earlier, this is something we're studying now. A lot of our models we've run out to a duration of 30 days. Now we're embarking on studies where we're treating for an extended period of time with the KRAS clinical candidate. I will say that in some cases we're out past 6 months and we are just starting to see maybe some signs of onset of resistance. And this is pretty reminiscent with some work we had done earlier, well, actually in a past life at Pfizer and otherwise, to try to understand resistance to EGFR inhibitors, ALK inhibitors, and other targeted therapies. Those often took greater than 6 months to start seeing onset of resistance. It appears pretty similar so far with KRAS. And we will have a series of models where we try to demonstrate resistance over time. In the models where we have observed resistance, we do not yet have any evidence that secondary mutations have popped up as a mechanism of resistance. And actually, we're still interrogating those models from a molecular perspective to see if we can get a better handle on whether there's other acquired genetic events, for example, those that may bypass KRAS signaling that would be responsible for resistance. But at this point in time, we have not identified any active site mutations as the key resistance mechanism.
Yeah, and Gina, also, I think maybe part of your question is, do we see G12C arising as a secondary mutation to like other driver mutations like EGFR or others? And in that, we have not seen that. You see very rare cases of overlap between KRAS G12C and any other driver mutation. So we think that's a very rare event, and that I don't think there's been any study of EGFR or other inhibitors with long-term treatment where you see KRAS G12C develop secondarily.
Mm-hmm. Okay, that's very helpful. And then I think another very technical question, it seems like KRAS inhibition works better in the 3D culturing than the 2D culturing. Do you think this is KRAS specific or just 3D culturing system is simply easier to show anti-tumor activity?
Yeah, that's a pretty fascinating question. I think it is fairly unique to KRAS. Have done some work in previous, either there's published work, have done some work in a previous life as well, where we've looked at the relationship of 2D versus 3D. And it often turns out that the 2D condition is more sensitive to, for example, chemotherapeutics that disrupt cell cycle and to a lot of receptor tyrosine kinase inhibitors. And I would say this is fairly unique for KRAS versus those classes of drugs. Why that is, I think we still probably don't fully understand. But a couple of possibilities and opportunities to study include, and in three-dimensional culture versus two-dimensional, there's more likely cell-cell adhesion molecules that are engaged, and it's well known that these adhesion molecules will sometimes complex with the upstream receptors and will complex with KRAS signaling complexes. So it's possible that these cell-cell adhesion molecules drive signaling flux through KRAS to ERK and otherwise, and further engage KRAS even when KRAS is mutated to drive signaling flux. Secondly, it's known that KRAS is a membrane protein, and now the group out of NCI has done a lot of work looking at KRAS membrane dynamics, and it's clear that KRAS is mobile in the lipid membrane when it comes to signaling, and suspect that there's something about the way that the membrane is curved in the 3D format that enables KRAS, kind of signaling dynamics, fluidity, and otherwise that would not be present in the two-dimensional cultures in which cells are more flat. So this is fairly unique to KRAS. We're not entirely sure why. We have some speculation on why that is. And would finally say that we've looked at this whole 2D growth versus 3D growth versus response in a mouse model, and that the 3D cultures tend to correlate much better with the response to in mouse models relative to the 2D culture. So we believe that this is a relevant model system to study KRAS biology in response to inhibitor.
Mm-hmm. Thank you. Very helpful. Now maybe we move to specific questions. We've seen the chemical structure of AMG 510 at AACR like a few weeks ago. So anything surprising when looking at the chemical modification of a 510 compared to its precursor molecule?
Yeah, I would say we were probably pretty close to being able to predict what Amgen's structure was. We've kept a close eye on Amgen. We have for quite a while believed that if we were going to advance a program to the clinic, that they would be critical competitors. I think the only surprise on their molecule was that there's a 2,6-fluorophenol that points to the aspartate 69 pocket, and we were a bit surprised to see that the molecule had a phenol. But suspect that there's a reason that Amgen selected that particular compound. So really that was the only surprise to us. Otherwise, the binding mode and properties of the molecule were pretty much as predicted.
And the reason for the surprise about the phenol is that it could lead to secondary metabolism. Right?
Yeah.
So that's the reason why we thought it was a little unusual.
Right. And it could just be that Amgen selected that molecule because it's sterically hindered and there is no secondary metabolism. But, it's certainly something, we'd like to understand our competitors. We'd like to study their molecules and understand what relative advantages we might be able to leverage in terms of development as we see their clinical data will likely come out before ours.
Mm-hmm. Can you help us understand a little bit more of the secondary metabolism?
Sure. There's a handle on certain ring systems, kind of aromatic ring systems. And if a hydroxy group is present on that ring system, it can be susceptible to secondary metabolism, most usually through glucuronidation, but also sulfonation. And this is one way that a drug can be cleared from patients. It essentially makes the drug more soluble and allows it to be cleared that way. However, not all phenols are subject to secondary metabolism. Sometimes they can be oriented a certain way that protects from that. So that would be something we would probably study here in the lab to understand better.
I see. So, Amgen's 510 also seems to have pretty short half-life. Do you think that that's also partly related to the secondary metabolism?
That's hard to say, but what I would say is for our molecule, we believe one advantage is our long predicted human half-life. So I think we've previously said or advised that our predicted half-life is somewhere between 20 to 30 hours in the clinic. Now, we haven't disclosed clinical PK data, but I think in a general sense, what I can say is that we're pretty happy with what we see in the patients so far. We suspect, based on all the preclinical modeling, that needing to cover KRAS entirely for the full dose interval is going to be important. And even though the protein half-life may be around 24 to 48 hours, we note that upon repeat dose administration of KRAS inhibitors to KRAS-bearing tumors, that what happens is there are several pathways that get activated that are designed to inhibit signaling flux through ERK. These include the dual specificity phosphatases, especially DUSPs 4 and 6. These include Sprouty and Spread family members, as well as the ETB transcription factor family. Once KRAS is inhibited, all these pathways get downregulated, and the cell becomes then conditioned to want to reactivate ERK aggressively if KRAS inhibition disappears. And we've seen that during dose administration studies over different regimens. So we believe that if we can cover or fully inhibit KRAS for the full dose interval, that's going to be important. Even during a 24-hour half-life or turnover of KRAS protein, if there is even 10 or 20% recovery of KRAS within the dose interval, we found that is a factor that may be associated with rebound or resistance. So, are really kind of riding on that this, long half-life is going to be important, and this is something we're really going to try to exploit in clinical trials.
Okay. I'm just following your line. I think the Amgen basically, they have a short half-life based on the animal model, like 30 minutes or 40 minutes, depends on model. And then also DP cycling is about 12 minutes. Half-life you mentioned about like 22 to 24 hours. So do you think that will be sufficient for once daily dosing?
We do think it'll be sufficient for once daily dosing, but it's certainly something we will study in the clinic. We plan on for us.
For us, yeah. I think she was asking about 510.
No, like
about
for Amgen's, yeah.
Oh, for Amgen's molecule, yeah.
Yeah.
I would say that we'll be watching at ASCO to see what the half-life really looks like. It would not surprise me to go to them to go to a different dosing regimen like a BID regimen, but we really just don't know at this point what their plans are and what their clinical half-life is. I'd suggest, based on the mouse models, that it's probably more in the 8-hour or under range, but just that comes with a big caveat. We haven't really studied it in detail.
Okay. Maybe one related question. You mentioned, in the human, you expect your compound will be 20 to 30 hours half-life. How's that translate, or is that similar in the mouse, or the mouse will be different, the half-life for the compound you enter clinic?
Mice are usually different. Usually as you go up in body surface area from mice to rats to dogs or primates to human, the half-life generally increases. Now, of course, this is also dependent on human metabolism versus the other species, which are other factors that can influence the half-life. I think one thing we're pleased about for our molecule is that, the half-life for our molecule is also shorter in mice. It's shorter in rats, but it's the most stable in human hepatocytes. The strongest metabolism was in human, and human also, of course, has the highest body surface area. So this translates to a nice long half-life based on our PK/PD modeling and empirical PK data at this point in time. Looking at Amgen's presentation, they didn't fully disclose their metabolism data, so it's not easy to predict exactly where the half-life of that molecule would show up, but suspect that we'll learn more about that here coming up in June at ASCO.
Okay. So when you say in a mouse will be shorter, I don't know if you can share with us, if your human is 20 to 30 hours, are we talking about hours? I think the precursor compound in mouse you had was 1.2 or 1.3 hours half-life. Is that also falling to the similar range? When we're talking about the compound you enter clinic, the 849 compound, the half-life in mouse?
Yeah, the half-life in mouse, rat, and dog is longer for our candidate than it is for the precursor molecule. And we specifically engineered the molecule to have that particular property of high metabolic stability and longer half-life.
Okay. And then, given also irreversible inhibition of the G12C, so do you think, the therapeutic outcome to be driven mainly by Cmax or area under curve?
We've studied that particular piece in detail. So we've looked at, again, mostly in mouse models, dosing twice a day, once a day, every other day, every third day, once a week, continuous infusion, and then mapping the parameters that we observe over each of those schedules back to the degree of anti-tumor activity and target inhibition that we see. And AUC or C average was the most closely, probably by far the most closely matched PK parameter to activity. So, it clearly wasn't Cmax. To me, this is not a hit and run target. It's a target that requires continuous coverage. And if the protein is turning over during the dose interval, it's likely that being able to cover at an average level is going to be important. I may have been a little bit surprised that it wasn't more C-min driven, but anyway, we took a very empirical approach and believe that average is going to be the way, or AUC is going to be the way to model our compound, and all of our projections in our clinical trial are based on that parameter.
Okay. Also, I think you mentioned quite some parts already, the differences between 510 and 849. So wondering if you can, touch upon a few more aspects like the chemical structure and a half-life you already mentioned, and then PK/PD a little bit. Can you share with us, how do you see the comparison 849 compared to 510?
Sure. Starting with binding mode, they have distinct binding modes. I'd say our molecules likely directly interact with His95. Theirs have a pi stack. Ours has a hydrogen binding interaction to His95. Theirs has a pi stacking interaction and pushes His95 to an out level. I think that's the only notable difference about binding mode. We don't feel like that difference in binding mode is going to translate to anything notable in that both of these bind to the inactive GDP bound conformation and are subject to the same changes or dynamics in GTP cycling. I think the molecules are both potent. They're going to achieve target potencies in humans, most likely. I'd say, where we feel the best about our molecule is, again, the nice, long half-life, the high volume of distribution. We think our molecule is going to penetrate very well into tissue. And then finally, the strong metabolic stability. We feel like, the properties of the molecule, we're not going to be very susceptible to either oxidative or secondary metabolism based on our preclinical studies thus far. That triumvirate of properties we feel like is our most strong competitive advantage at this point in time.
Mm-hmm. And I think some of the mouse models were the same, in both yours and Amgen's studies, like MIR-PACA2 or in NCI H358. So others are pretty different. So how do you see the anti-tumor activity of the two drugs, in the same model?
Yeah, hard to say because we don't have a head-to-head comparison, but, we did note that if you're looking at the MIR-PACA or H358, we feel like we're able to see a bit stronger anti-tumor activity. We also did note the PDX models that were part of the presentation. When we publish on this, you'll see that the lung PDX models that we included in our studies, we see, pretty comprehensive evidence of tumor regression, in some cases complete response in those models. Again, hard to say. These are, they didn't note what those models were or otherwise. So there may be some differences there. I suspect probably what's behind those differences is what we just talked about. The half-life and target coverage to maximize anti-tumor activity within the given dose interval is very important. And we've studied this in detail over various dose schedules and believe that half-life is going to be key. Now, note, of course, these studies are in mice and not in humans, so it's hard to draw major conclusions of superiority in activity in mouse. But again, we're pushing for the nice long human half-life and full target coverage and believe that that's going to be an advantage.
Yeah, and one other thing, just to mention, I don't know if Jamie mentioned it, the volume of distribution is greater for 849 than what we saw for 510, and that could also help in terms of accessing the target and resulting in higher levels of inhibition.
Mm-hmm. Okay. So just want to reaffirm, the M genes, I think the 2 PDX model to me was not super impressive. They only show tumor regression. They did not show tumor regression, only show the slow growth compared to untreated. So, and they use pretty high dose, like 100 milligram per kg. So the underlying, the reason you can think of is that, the thoughts you mentioned, is that mainly related to, say, half-life tumor distribution that make a difference. Maybe your compound, like 849, may show different efficacy or activity.
I think so, and this, of course, is speculation because we haven't studied, but I would suspect, based on everything that was shown, that AMG 510 is, fully or near completely inhibiting KRAS for the first part of the dose interval, but it's recovering during the second half. And that recovery during the second half of the dose interval is something that we have observed in earlier compounds to be associated with submaximal antitumor activity.
Mm-hmm. Okay. And then when will we see the 849 preclinical data?
We haven't really disclosed when that is, but we are putting both publications and presentations together, likely for later this year. We may even try to coordinate the disclosure of our preclinical and clinical data at the same time.
Yeah, that's a possibility. I think that's something that we're looking at, Gina. I think we've told you before that our patent doesn't publish until May, so we didn't want to do anything prior to that. So it'll be sometime after that.
Mm-hmm. Okay. And then also, I think we likely will see the MG510 phase 1 data likely at the ASCO. So what will be, read through to Mirati, and then what will be the moving parts that investors should pay attention to?
Yeah, I think really you've touched on a lot of those already, right? In thinking about the comparison of the preclinical data, and that we obviously feel strongly that full inhibition of the target over the entire dosing period is important, and we think that we can achieve that. And so we would hope that that'll translate into a better clinical outcome. So what I would say is that we believe this is an important driver mechanism, that you will see activity with the Amgen compound, although we have no direct knowledge of that, so it's hard for us to know exactly what they're going to show or how many patients they have. So it's really tough, as you know, in phase 1 data. You have patients treated at differing doses, and that can affect response rates, things like that, different tumor types. So there's a lot of variables here. But I think that, seeing activity there would be helpful. It's just proof of concept that there is activity in patients. But I think, we feel good about 849 and that we may be able to do better in terms of clinical activity than the Amgen data. So that's what we're trying to get across in at least comparing all of the data out there so far, which is all preclinical, that's the basis of why we think that.
Okay. Okay, that's very helpful. So, are we, do you, I think at the beginning you mentioned, Jamie also mentioned, certain response rate. We're talking about 60% response rate, durable 9 months. Is that the bar you were thinking, or what would be the initial proof of concept you think would be very encouraging, say from Amgen's data? Yeah.
Right. So I think we need to step back a second and look at this in 2 ways. And I think the simplest way might be to just talk about lung cancer first. And so if you look at certainly in our study, the patients are going to be second, third line, most likely, and I would suspect it'll be the same with their study. And in that setting, that we believe, and based on our prior interactions with the FDA, that if we had a response rate of 20% or greater, and those were durable responses and you had a good safety profile, that could lead to an approval, right, in that setting. So we think that that is the regulatory bar. But again, going back to what Jamie said earlier, that probably the closest analogy we can come up with is BRAF-MEK combination in BRAF mutant non-small cell lung cancer patients. Those studies have been done. I think there's good data there. That's inhibiting the same pathway. You know, it's a different patient population because it's the BRAF mutants, but that in that setting you can see over a 50% response rate and good durability of over 9 months with the combination. So I think that gives us that, you know, that's a good sort of benchmark to think about. We don't know exactly, obviously, until we get the data, what that response rate is, but I think, you know, as long as we're above the regulatory hurdle, we're in good shape, and that anything above that just makes it easier, right, to go forward as a single agent. And the higher the response rate, the easier it is to go into earlier lines of therapy as well. Now, we are also, you know, Jamie noted, and we've talked a lot about combinations, and we do believe that combinations are also an important part of the program, and it's something that we've built in early on to the program. And even in this initial phase 1 study, we are doing those combinations, which we think are most interesting. And Jamie mentioned things like the HER upregulation. We're interested in SHP2, you know, certainly with PD-1 combinations, and he mentioned CDK. So those are things that we're starting very early in the program as well to broaden the program, to increase the response rate even further, and to get longer durability, potentially to combat resistance. So, you know, we're trying to take a comprehensive approach here to inhibit the pathway as strongly as we can. So does that help answer your question?
Yeah, no, that's very helpful. That's very helpful. Okay. And we will certainly touch upon the combination later. Okay. I do have an email question now asking about the ASCO presentation on Saturday. The title was out, and basically phase 1/2 multiple expansion cohort trial of 849 in patients with advanced solid tumors with KRAS G12C mutations. What will be shared for this presentation?
Yeah, that is just trials in progress. So okay. as the rules are pretty strict about those, ASCO's quite strict. And so that is really just to describe the design of the trial and to go through the details of the trial design, but not to present data on the clinical outcome.
Okay, that's perfectly to my next sets of questions regarding trial design. So we know that Amgen, 510 has they already started last year August, and your phase 1 started in January this year. You do have a 1+1 trial design versus traditional 3+3 design. Can you walk us through your dose escalation scheme?
Sure. So let's start with just the dose escalation scheme. So we started at 150 milligrams once a day, and then the escalations were 100%. So 150 to 300 to 600 is the cadence of how it's done. And then also, once we've passed the prior dose, the safety portion, and then dosed a patient at the next higher dose, the earlier patients in the earlier doses can up dose to, or intra-patient dose escalation to the next level. So that's how the study works. If we see any toxicity, then we add additional patients, and we can expand at any time when we see that we're covering the target. So we have good evidence by PK that we believe that we're hitting the target completely, and that we start seeing some sign of clinical activity as well as good safety profile, that we can expand any of the cohorts. At the same time, we can continue dose escalation up to the maximum tolerated dose. So it's, I think, a creative design that allows us to answer a number of questions, to find the right dose, because we want to make sure that we go into the next phase in escalation at the right dose, to get the best chance of seeing clinical efficacy. And then once we do see activity, we can expand this study and single agent all the way potentially to a registration study. So all of the parts of the study are designed to facilitate moving as quickly as we can.
Chuck, can you walk through like a specific timeline? Like say if you dose the first patient 150 milligram, how long does it take? Usually, based on what criteria you dose the second patient, and then how soon you also scan the patient?
Yes. So the way it's done is that because this was first in human and it's a novel mechanism, we actually did a single dose, and then we get the PK from just a single dose. Then we do multiple dose, and then we get that PK information as well as safety to decide if we go up to the next dose. That process takes about 4 weeks per dose, roughly. Now, there's a lot of other things that can happen. Patients can fall out, that sort of thing. But generally, it's 1 month per dose level, if everything goes according to plan. And then, as I said, it goes up to the next dose level, and that dose is the same thing. You do a single dose, then you do multiple dose, and then you can decide if that initial patient moves up. In terms of the scan frequency, scans are done every 6 weeks. So we arbitrarily designated 3 weeks as the cycle time, and so it's every 6 weeks or every 2 cycles that patients get scanned, and that's continuous. So every 6 weeks for the duration that they're on the study, they get a scan.
So that means you actually could likely dose patient up before you actually get the scan, right? So how would you decide, after the scan, how would you decide the dose, intrapatient dose escalation?
Yeah, so as long as there's a good safety.
Sorry, like within the same patient.
Intrapatient, yes, exactly. So as long as the patient is tolerating the dose well, they could escalate to the next highest dose, even if it's before they get their first scan. So that can happen because it's really just a safety determination. But, in parallel, we're doing the scans along the way, right? So we'll be getting that information as we go. And then it helps us to decide, what the next dose level should be, as well as, whether or not we should expand at that dose.
Okay. And then under what condition that you would not dose escalate?
Yeah, so it would go back to the basic, characteristics of a phase 1. So if we see dose-limiting toxicities, we would expand the cohort, and then if we see a second dose-limiting toxicity, then you would stop and declare the prior dose as the maximum tolerated dose. So that's it. In that sense, it's very typical of other oncology studies. So, as we've talked about before, and something we really didn't touch on yet, we saw a very good therapeutic index in the preclinical studies. So the safety profile of these agents looks pretty good, and at least from the sort of gestalt that we've heard from Amgen, they're seeing the same thing. So I think that the tolerability is good, and it could be an interesting case where we actually choose a dose below the maximum tolerated dose as long as we're fully inhibiting the target, which is why we designed the study this way, to give us that flexibility. So it's something that I think is interesting and will be obviously interesting to talk about when we have the data presentation later this year.
So if patient you dose up, say from first dose to the second dose, the same patient, but then the scan comes back, and if they show, say, stable disease or progression or response, like how would you choose the next dose for that patient?
Yeah, so again, it's a little complicated for those intra-patient dose escalations. It's cleaner for those that start at that dose and then continue. But certainly if you see that it's well tolerated and the patient has some activity, whether that's some degree of tumor regression, that you could continue them on it's partly the patient and the investigator could decide to continue on that same dose, or they could continue to dose escalate. And as we said, we believe strongly that the higher doses, the complete inhibition of the target is important. And so we think it makes sense to continue the dose escalation, even if you start seeing some activity. But that is up to the discretion of the patient and the physician at that point if they see activity.
How do you define activity? Will stable disease consider activity?
You know, if we see, usually the way I think about it is if there's tumor regression, I mean, and as you know, stable disease includes those patients that have some degree of tumor regression. You know, if they have slowed growth, I tend to consider that less impressive, right? And that it would make sense to continue dose escalation. And even if they have less than a partial response, I think it's worth going up to the next dose. So as long as there's room for improvement, essentially, I think it makes sense clinically to go up to the next dose.
Okay. What about patient had a progression or like the tumor grow beyond 30%? Will you continue to put patient on drug, dose escalate, or those patients will become off study?
That's a case where there's some degree of discretion on the part of the patient and the investigator. So as long as they're still doing clinically well and they show what the investigator thinks is some benefit from treatment, they could continue on study at the next higher dose. But generally, these patients, I'm not commenting on this study, but generally patients, once they progress rather quickly. So they tend to come off study and tend to deteriorate. So it's less likely that would happen, but we could certainly consider it if there's evidence or in the investigator feels that there's benefit.
Mm-hmm. Is it fair to say, based on the timeline and your description, is it fair to say like now you dosed at least 3-4 patients now, and then get scanned maybe at least two patients?
So we haven't made any public disclosures of where we are. We were trying to resist, doing patient by patient updates, as I think you can understand. What I can say though is that this trial is going well, that we're not having any trouble finding patients, and that we're progressing through the dose escalation. So, we believe we're on schedule and on plan.
Mm-hmm. And I think, Chuck, last time when we talked, you mentioned that, the initial dose, 150, that would be equivalent to mouse like 10 milligram per kg dose. And do you expect this dose to be within the therapeutic window? And then what will be, based on the mouse data and human PK/PD data, what dose range do you think will be optimal therapeutic window?
Yeah, so I'll let Jamie, because a lot of this is extrapolation from the preclinical data, so I'll let him comment on this one.
Sure. Yeah, 10 mg per kg should start to enter the therapeutic range as a dose that would yield a partial response in sensitive models. And in fact, at this 10 mg per kg dose, or the 150 mg dose in human, we suspect that the therapeutic index is probably another 20x. So we can continue to go up without expecting to see signs of toxicity based on our preclinical data. What's optimal? We suspect to be able to start hitting that at what would be the equivalent of 30 mg per kg dose, which would be roughly the 600 mg dose QD in humans. So then we start to get into the optimal range. As Chuck mentioned and I mentioned earlier, we really want to be assured that we are completely inhibiting the target for the full dose interval in the vast majority of patients. So we would continue to, escalate until we're satisfied with that, or we see by protocol dose-limiting toxicity that would ultimately define our clinical dose moving forward. So we will be taking safety, PK, and efficacy data into account as we make those decisions.
Mm-hmm. So thank you. That's very helpful. So let's say safety looks very good, and what would be your criteria, say, for activity you have to see in order to start the expansion cohort?
Yeah, so there's two parts to that, essentially. One is obviously tolerability. So we would have to be well tolerated. The PK would show that we're at levels that we believe we're fully inhibiting the target. And then the third is some sort of clinical benefit. But again, we can start the initial expansion up to, say, 6 patients without seeing the first scans, right? So if we're seeing what we believe is the right dose and the PK looks good, the PK/PD looks good, then we could start, but we would start with something like 6 patients. Then once we see the first scans on those initial patients, we could expand further. We can go up to 40 patients initially, and then we would decide based on activity if we expand further than that. But I think that gives us a lot of flexibility to use all of the data, tolerability first, of course, then PK, and then efficacy together to make that final decision about what dose.
So maybe if I just only isolate, I know the other moving parts are also very important. If we're only focusing on the response rate, what kind of response rate will make you expand, say, from 6 patients to 40 patients?
It's difficult because 6 patients is a small number. So usually it's a sort of Simon design. So if you saw 1 patient, it would be at least 1 patient. You would be expanding further. But like we said before, we want to have at least something that we think is would be above the regulatory standard of at least 20% or higher. So that, the higher the better, obviously, but we would, in those first 6 patients, be looking for 1 response. If we have more than 1, obviously, we would activate and go to larger patient numbers faster.
Okay. And also, I know it is very early, lots of assumption involved. Just wondering, how many patients do you expect to have in your initial data presentation later this year?
Yeah, I mean, you can do sort of a calculation now that we started in January. But I think that it's reasonable to say that 10 patients at least by that time where we would have some initial data, PK, obviously, safety data, and some initial activity data on response rate. The durability, obviously, is a question that's going to take time to answer. Since we only started in January, if we present in the second half of the year, we won't have really long duration, but I think we'll start to get an idea of durability as well, but it'll be early for that.
Okay. And then regarding the timeline, previously you mentioned that, likely went long, but later was like triple meeting. Is that still the case, that, the likely triple meeting?
I think that's a good forum for this kind of data since it's a novel agent, it's early development, it's a good audience for it. It happens to be in Boston, which is convenient. So I think those are reasons why it might be, but we don't have a final determination yet of when that would be. But that's a reasonable meeting, I think, for this kind of program.
Okay. Any scenario you may present the data earlier?
We don't plan to. We think that it's wise in this case. We obviously have confidence in the mechanism. We are in good shape, financially. We don't need to do anything sooner. So I think that it's wise for us to have the most comprehensive data possible so that we can say something about response rate as well as durability when we do the first presentation and have a good understanding of the PK/PD profile. So that's why we're saying in the second half, and that's what we continue to maintain at this point.
Thank you. That's very helpful. Maybe, operator, let's pause for a moment and see if we have any questions from the audience.
Yes, ma'am. Ladies and gentlemen, just as a reminder, if you'd like to ask a question at this time, please press star and then the number 1 on your telephone keypad. Once again, that is star and then the number 1.
Okay, while we are waiting for the response, maybe I will ask one more question regarding the combination therapy. I think Chuck and Jimmy, you already mentioned earlier EGFR, the SHIP, CDK and PD-1, that could be the top choice, and HER as well. So I think there are from MG510 data, actually they show pretty impressive data with PD-1 and as well as the ERK inhibitor. So just wondering, what is your thoughts based on your data and the MG data? What will be the top choice for you for the combination going forward?
Sure. Yeah, I mean, I think starting with PD-1, we recognize that G12C is a transversion. It's present in smokers. They tend to overexpress PD-L1. And now, based on clinical data, it's known to be an immune-responsive population, but still a strong unmet medical need. There's a mechanistic rationale to combine KRAS inhibitors. Blockade of KRAS can upregulate MHC class 1 and perhaps class 2 molecules, augmenting antigen presentation. And they also control some downstream factors like VEGF-A, IL-8, and CXCL cytokines that may change the tumor microenvironment. We were also compelled and thought that the Amgen data was pretty neat. So this would be one of our, I think, top choices. We think that strategies like EGFR or a drug like afatinib or a SHP2 inhibitor are going to be important to combat all of this signaling pathway feedback and further dampen the signaling through ERK and block ERK flux. And we've seen these strategies work almost universally in mouse models to the point where models that are somewhat refractory are now thrown into partial responses. And some of our partial response models are becoming complete responses in combination. So we are very compelled and we'll be pursuing this as well. And as I mentioned, it's possible that CDKN2A is a resistance mechanism to the drug. We believe that this is readily addressable in this segment, which is basically around 15% of lung cancer patients, that this particular alteration may be addressable through that combination. Ultimately, it may turn out that we have certain segments of KRAS, like the co-mutation of KRAS G12C plus CDKN2A, that are addressable by one combination, but other segments may be addressable through other combinations. So we will be also subsetting patients as part of our combination strategy to explore that possibility.
Yeah, and I think the other one you mentioned was MEK as a MEK inhibitor. You know, we have done a fair amount of work with MEK and RAF and pan-RAF compounds in combination. We haven't seen really the kind of synergy that we did with these others, and we think that's because we're hitting the KRAS pathway so hard, and when you completely inhibit it, then it's less likely you'd see activity with MEK inhibitors, for example.
Yeah, and I think here I would just add, in some cases we've seen combination activity with MEK and broad-spectrum RAF inhibitors, but would just say it's not as universally active as it is with EGFR and SHP2 inhibitors. So we like the vertical pathway inhibition strategy, but blocking upstream of KRAS as opposed to downstream of KRAS.
Mm-hmm. Okay, that's very helpful. Operator, do we have any questions?
No, ma'am, there are no phone line questions at this time.
Okay, great. I have a few more questions. Maybe we can quickly ask about Citro combo. A few proof of concept data will be presented second half this year. I think some from you and then some actually from your partner at Beijing. So how much data should we expect to see, and then what will be the bar, for I think a few key indications to be considered clinical meaningful?
Yep. So yes, we are initiating the phase 3 study in this quarter. So that's great in non-small cell lung cancer, second-line non-small cell lung cancer. The combination data in other indications were, as you said, in the second half of the year. Certainly for refractory bladder cancer, we also had a couple of studies which I think will be interesting later this year. One in essentially neoadjuvant head and neck cancer and one in neoadjuvant renal cell. So those are 3 studies that we are conducting, and we would expect to have data from those in the second half of the year. For bladder cancer, we're thinking that we needed to see at least a 20% response rate. We're thinking we should have data on somewhere between 10 and 20 patients in the second half of the year. So to give us an idea of go, no go for that checkpoint refractory patient population. For the neoadjuvant studies, the main focus there was really on mechanism of action and understanding the mechanism of sitravatinib in that pre-surgical patient population so that we could get better idea of the PD markers and to correlate that with the preclinical data we've done. So that I think will be interesting and useful information that could help differentiate us from other RTKs, because we do think that the very potent inhibition of tyroxine and MER by sitravatinib is an important part of that mechanism. For BeiGene, they're doing a number of different tumor types, as you noted, in phase 2. We're talking with them. We have a very excellent collaboration with BeiGene, which is progressing nicely. They're enrolling patients. We don't know yet what their plans are for presentations the second half of this year, but certainly the enrollment has been good, and we would expect to have, a number of patients treated in these different indications. We just don't have all the details for you today. But we are looking forward to that. We think that hepatocellular, gastric, ovarian, all of those are interesting combination opportunities. They are also able to do first-line non-small cell lung cancer in combination with tislelizumab, which we think is a very interesting opportunity for us. So, though, it's progressing well, and we'll have more information to share later this year.
Thank you. And I do have one email question regarding the partnership, the G12C, the KRAS G12C and G12D, the licensing deal with Array. If you can elaborate, what is the royalty and the milestone?
Okay. I'll let Chris answer that specifically, but did want to mention, and we should mention that we've had a very good collaboration with Array in this program on G12C and G12D, which is ongoing, and that we do have exclusive rights to both of those programs. And then Chris can comment about the details.
Sure. So very, very shortly, this was an FTE-based discovery relationship. Once we progress beyond that, we have milestones until the drug is approved and some sales milestones. We haven't disclosed the amount. And what we've said about royalties, they are tiered. So the actual royalty rate will be dependent upon overall annual sales levels, but you would expect that we would expect the effective royalty rate to be in the mid to high single digits, and if things go really well, it could move into the low teens as well, which we hope it does.
Okay. So what about the development milestone and the sales milestone? If you can give a little bit more color on that. I think that there's some numbers out there that talking about $300 million. I'm just wondering, what is the numbers that we are referring to?
No, that is correct. That's the total amount is in that range, and what we've been able to say, because we haven't disclosed all the specifics, neither us nor Array, but that more than a majority of that is associated with the sales milestones.
Okay,
the rest
and that's it.
are over development and ultimately approval.
I see. So is the 300 range of $1 million, is that for one compound or for both compounds?
That's correct. One is for the G12C program and the other is for the G12D, and they're both the same amount.
Okay, same amount, around 300 something million.
That's correct.
The sales milestone. Okay.
Primarily sales.
Well, thank you very much. I think that this has been very productive call and then really helpful. Thank you for your time, and also I wanted to thank everyone dialing, and well, happy Easter.
All right, thank you very much. Appreciate it.
Okay, thank you.
Thank you.
Have a great holiday.
Bye.
Ladies and gentlemen, this concludes today's conference call. You may now disconnect.